Well, good afternoon, and welcome to our Asthma Grand Rounds. I'm Chris Fanton. It's a pleasure to welcome you, including those of you who are joining us by live webcasting today. And I'm pleased to introduce our speaker, Dr. Alexey Fedulov, who's a member of the Pulmonary and Critical Care Division here at the Brigham and Women's Hospital, and also a member of the Department of Environmental Health at the THJ and Harvard School of Public Health. And his laboratory investigates the epigenetic determinants of inflammatory and immune disease processes, as well as the role of DNA demethylation as a potential therapeutic modality. And so we're very pleased to have him here to talk to us about asthma as a transgenerational epigenetic disease. Thank you, Dr. Fedulov.
Well, thank you, Chris. Hello, everyone. It is a great pleasure and an honor to talk here today. As Chris said, my talk is about transgenerational mechanisms of inheritance in asthma and about epigenetic mechanisms in this disease. And as an outline of the talk, I will first start with introducing the idea of why we need to consider any new paradigm at all in asthma, and in part, why do we consider epigenetics as part of a paradigm. I will give then a brief intro about the epigenetic mechanisms and what is transgenerational inheritance per se. And then we will look into some evidence that supports this theory, both in humans and in mice. And in the end, I will shift gears a bit and talk about epigenetic engineering or epigenetic editing as a way of what we can do about all these concepts and how we can move these towards new therapeutics, perhaps. So in the history of asthma, as collected in this slide, we've been generally able to come up with therapeutics that are mechanistically based. And in fact, we know quite a bit about mechanisms of asthma occurrence and development, and we recognize that asthma is a chronic inflammatory disease of the airways. But before we knew that, we learned about one of the first things perhaps was that asthma includes bronchial constriction, and we use bronchodilators for treatment. Asthma includes mast cell mediators, and we might use membrane stabilizers, and with lesser use, but in general in allergy we use histamine blockers. We've then learned about immunoglobulin E and the B cell production and in general inflammation in the airways, hence we use anti-inflammatory drugs like steroids. In the later years we learned about the disbalance between T helper type 2 and type 1 environment. And there are some experimental agents like CpG oligonucleotides, and there's a debate on the role of DPT vaccine in skewing this aberrant T helper 2 balance away towards less allergic Th1 responses. Most recently we learned about the role of dendritic cells and antigen-presenting cells in general in the decision-making point when they sort of decide to present a particular antigen to T cells and induce this aberrant inflammatory immunity that we see in asthma. But in fact, none of these are causative, and none of these allow us to work on the etiology of disease and figure out why in some people asthma occurs and why in some doesn't. And here I attract your attention today, and I hope it's a time well spent to consider a relatively novel paradigm that DNA methylation, histone changes, and other epigenetic machinery is involved in asthma etiology. So certainly asthma is a multifactorial disease. It involves interaction of genes and environment, and most recently epigenome, and they all interact to affect children or a fetus or a female organism and to some extent male organism, and they sort of contribute to, as components to the origin of asthma. And this concept doesn't need too much argument because it can even be reviewed in one study. In this paper by Dr. Salam et al., for instance, they looked into a haplotype of a promoter of a gene and a methylation in that same promoter, and they investigated in people the potential outcomes and the factual outcomes of exposure to particulate matter. And their target gene was nitric oxide synthase, and the outcome they looked for was exhaled nitric oxide. So in fact, they found that both the haplotype of a nitric oxide synthase gene and methylation in its promoter both contribute to exhaled nitric oxide when the children were exposed to particulate matter. So this concept integrates data from genomics, epigenetics, and environment. But for the purpose of today's talk, we'll focus on epigenetics, and one reason is that genetics and environment alone cannot really explain the overwhelming growth of incidence of asthma over the last couple of decades. Often there are no family history in asthmatic patients, and if in some of these studies, when they do genetic profiling, they find no known SNPs or other genetic code changes. Well, there is a need to focus on an unknown component and try to get the most of it. And one way to focus on this component, particularly I'm hinting towards epigenetics, of course, is to try to use experimental models to dissect this epigenetic studies. So by way of introduction, epigenetics entails changes in the DNA and chromatin that occur without a change in the DNA sequence. These involve DNA methylation, which is an ability of cytosines in the DNA to be bound by methyl groups, and thus in accumulations of cytosines called CpG islands, this can affect transcription. These changes involve modification of histone tails, a phenomenon known as RNA interference, for example, microRNA, and some other mechanisms. And there's more than 100 various biochemical, if you will, modifications to the chromatin that can affect its function. So I like to use this comparison in virtue of my recent flight school experience, that if this is an airspace above Boston and the bird's view on it, you see the underlying landscape and there's freedom to move in any direction if you are a bird. But if you're a pilot, there is an overlay that looks something like this, with a map of instructions on where you can go and where you cannot go and how it all works and organized. So in a similar way, epigenetics is a markup on the DNA code that tells the cell how to operate with its genes. And in fact, it's a simple biological concept, but we don't really think about it much. One that involves the idea that in a eukaryotic organism, all genes in each cell actually are the same, and they comprise all the genes of that organism's genome. But nevertheless, the difference in structure and function of cells is tremendous, and it is explained, in fact, by epigenetic, as we call this type of mechanisms, by which the cell silences unnecessary genes. And there are quite detailed studies now that sort of look into this landscape of epigenetic development from, let's say, a stem cell or a pluripotent precursor to partially committed and to completely committed cells. And if you do methylation analysis, you will find that the repertoire of genes that is expressed in the cell changes as the cell goes in this development. So some parts of the epigenome are hardwired in a cell type, and as the cells mitotically divide, they look a lot like each other, and this is mediated by the fact that their epigenome is restored after division, and perhaps the mechanism is not fully understood. Some changes, however, can show plasticity in response to some change of environment, and this is perhaps a way of cells to respond to and adapt. The alterations that are induced in the cell, for instance, in a cell culture, if you do it in an experiment, they may or may not persist, and it's one of the challenges in epigenetics where we don't know, for many cases, when we induce this kind of change, let's say in DNA methylation, how long it will persist. But some persist, and it's true for both quiescent cells and mitotic cells. But most interestingly for our purpose today is that some alterations can be inherited, particularly inherited meiotically. So inheritance of epigenetic alterations is one of the basic sort of culprits in the field. And this idea that epigenetic processes are not fully erased during gametogenesis has come up in the last couple of decades, maybe the last decade. And it generally counters to what was previously believed, and so it's fair to say that there are some controversies and debate about it. But it appears that there is growing evidence that for alterations in the organism's epigenome, for instance due to an environment, there are some changes that can be meiotically inherited and some don't get inherited. So the mechanisms of these are poorly understood, and one of the things that is exciting and not quite clear yet is whether transgenerational inheritance is mediated by the same mechanisms as the mitotic inheritance in a cell population, for instance. Another question is whether metastable epialleles contribute to the transgenerational inheritance. So an epiallele is a stable allele that can exist in more than one epigenetic state, for instance, methylated and silenced versus unmethylated and open. And the metastable epiallele is one that is variably expressed in genetically identical individuals due to epigenetic modifications. And this is reprogrammed during early development. And there is some evidence for metastable epialleles that how they contribute to generational epigenetic inheritance. One example is seminal studies by Randy Jordal and colleagues that look into the AV metastable epiallele, and it stands for agouti viable yellow. This is a gene that encodes coat color, and coat color in animals is a genetic principle, genetic feature. So in let's say an AV normal mouse, the coat color is yellow. And as it turns out, this locus is particularly responsible to methylation changes in its promoter. And if you give a folate, which is a methyl donor, if you give a folate supplement in a diet, these mice will change the coat color because the function of this gene changes and become pseudo-agouti or mottled. But most exciting about these findings is that this gets inherited even though the offspring of these mice don't get any folate supplement. And in fact, the ones that are born from yellow mice are yellow, and the ones born from mottled are mostly mottled. But getting closer to asthma, there are studies that look into this. And one example is a study by Hollingsworth, where they used, again, folate diet or another high methyl donor diet to try to promote asthma in the offspring, and it appears that it worked. So in this comparison, on the upper right, you're looking at the airway responsiveness to methacholine, and as you see, it is elevated in the offspring of mice that were fed high methylation diet compared to low, and it's also true for levels of interleukin-13 and eosinophilia. So it appears that some of these epigenetic changes are inherited, and this was an example of intragenerational inheritance, meaning the first generation. But even more interesting concept is transgenerational inheritance. It is defined as germline-mediated inheritance of epigenetic information between generations in the absence of direct environmental influences that leads to phenotypic variation. So basically, transmission of this epigenetic information to the next generation will affect the epigenome of the developing embryo and its embryonic stem cells, and in a way that the subsequent somatic cells and tissues that are generated in this fetus will have transgenerational change in the transcriptome. So when we talk about these concepts, it kind of reminded me of something, and it is fair to say, and let's do this in a style of Jeopardy. So what is this biological phenomenon that there's a change in the environment that brings about a change in needs and behavior and function of the organ, and therefore it brings this change in the organ usage and the development of function, and then can transmit to next generation and thus mediate changes in the population, right? And the answer is, what is Lamarckism? So we're really living in a sort of an era of epigenetic Lamarckism, where there's a rehabilitation of this concept, because some studies actually show, particularly behavioral studies in rodents and birds, that, for instance, food behavior or ability to solve maze is inherited. And for instance, offspring of rats that were trained to solve maze 5 times quicker can also solve this maze much quicker than the offspring of control rats. So there seems to be some sort of a molecular basis now for this type of findings, even though they are rare. But it's fair to say that there is something to look into in terms of evidence of transgenerational inheritance, and we're about to do this. So there are various possible scenarios of transgenerational inheritance. So if you look at the lower square in this chart, you will see that on the left, the sign or the feature may disappear over generations. In the middle, you will see that the feature may consistently appear in every generation downstream. And on the right, there is a sort of veining of the phenotype, or occasional appearance and occasional loss. And in my opinion, the last one is the more characteristic for epigenetic inheritance. But one interesting difference between intergenerational and transgenerational inheritance merits discussion, and it has something to do with pregnancy exposures. Typically in experiment, we look into exposures during pregnancy because it seems to be this window where the changes are easier to induce, perhaps. And in people also, pregnancy exposures, we know, matter more sometimes. And so in fact, the concept is that if we're talking about an exposure of a pregnant animal or a pregnant person, the fetus already has embryonic cells in it that would give rise to progeny of F2 generation. So to avoid the risk that our exposure is actually directly toxic to the cells and not inherited at all, and that would be what we're looking for, we need to follow to at least F3 generation to say that this is somehow related to the epigenome and perhaps in the gametes. And there are, in fact, a few studies that do this in an experiment. There's one or two that I know of asthma, and these are by Dr. Rehan, and they've traced 3 generations, well, to F3, down the offspring of rats that were exposed to nicotine during pregnancy, and they found that this exposure actually predisposes to various asthma-like features in the offspring when they're challenged with allergen, for instance, airway hyperresponsiveness shown here on the upper left. Interestingly, though, this was also linked to epigenetic changes in DNA methylation and histone acetylations shown in the lower right. And this was seen in both lung and in the gonads, suggesting that germline tissues, at least, are involved and epigenetic change can be detected there in association. So in this wonderful review by Martel Hilkema last year, there's a summary of evidence for pulmonary disease, particularly for asthma and COPD. And it would appear that there's a number of agents in the left column here that have been traced to several generation downstream effects. They involve tobacco and nicotine components. They involve allergens, pollutants like BPA. They involve bacterial and fungal components and nutritional factors like high methylation diet and stress and others. But really only 2 of these papers, and I've just shown you one of them, looked into F3 generation inheritance, as we would suggest being truly transgenerational. And there's a couple of other studies in animals unrelated to asthma that show vinculazine or dioxin effects are associated to disease, with disease in 3 generations down. So getting to this concept that asthma could be an epigenetic disease, or at least in many cases has a strong epigenetic component, well, what would be the definition of this? And there isn't one really, but if we try to come up with one, we would say that to be able to say that, we look into aberrant epigenetics in this disease. Then we would, let's say, hypothesize that there could be an unequal maternal versus paternal influence, and this is frequently seen in epigenetic phenomena. We would look into heritability and particularly interested in transgenerational transmission of this phenomenon. And we would also try to have some kind of experimental intervention that could affect epigenetic pattern and abrogate or prevent this transmission of disease. So this is a call for studies, if you will, and let's try to look into some more details of how this is done. So firstly, in people, this is a great study by Dr. Runyan et al. And they've looked into a cohort of genetically identical twins where one of the twins had asthma and the other didn't. And they in fact found a change in methylation of a FOXP3 promoter, which is an important gene in regulatory T cells that is anti-asthma working. And they found that methylation is significantly elevated in the twins with asthma as opposed to healthy. And it did correlate well with the expression of that factor as well. So this suggests that we can go beyond genes and perhaps we can go beyond environment to suggest that epigenetics can be quite important in the causality of asthma. Other studies have made similar findings. Dr. Miller's group looks into maternal smoke and childhood asthma, and they also find aberrant epigenetic marks in the diseased group versus healthy. It is known that highway exposures and to exhaust, and particularly diesel exhaust is playing a significant role in this. This type of pollution factors play a role in changes in epigenetics, for instance, detected in people who live closer to highways. And there's also a lot of data, even collected over the many years past, that people who live next to highways have higher likelihood of asthma development. So for parental story, there seems to be a consensus that maternal history of asthma is more likely than paternal to affect the risk in the neonate, and at least in these studies that I reference on the slide, you can see that maternal influence was more significant than paternal. And while in people this could be multifactorial, we're trying to make experiments to dissect this effect. And we've actually created a model of maternal transmission of asthma in mice. The model was created by Dr. Kaoru Hamada, who worked with Les Kopzik some decade ago. And in this model, we induce ovalbumin allergy and asthma-like phenotype in female mice. And then we mate them and control mice with a normal male. And then we test the offspring in a single injection, sort of a low-dose allergen protocol for asthma-like responses. And if control neonates show no or minimal asthma features, the neonates born to asthmatic mothers with the same exposure show airway inflammation, infiltration, eosinophilia in the lavage, elevated hyperresponsiveness in the metacolon test, raise of cytokines like IL-4, 5, and 13, increases in immunoglobulin E, and other factors. So in a variation of this model, we expose pregnant mice to environmental particles, for example, diesel exhaust particles. And then we test the neonates similarly, and we find that in these cases there is, if you look into comparison, there is also a transmission of a phenotype to this F1 progeny, and we can see elevated BAL cell eosinophilia counts on the right. This is true for diesel particles, titanium particles, and carbon black, and elevation of airway metacolon, airway responsiveness in the metacolon test as well. So this suggests that pregnancy predisposes towards inflammatory responses, and the outcome of such inflammatory responses can be a risk of asthma in the neonate. Well, we started looking on the neonatal immunity and tried to find out what is it that is changed in this neonate that is born to asthma or particle-exposed mother. And we made a series of adoptive transfer experiments of immune cells from the susceptible offspring before allergen exposure to a control recipient. And we've tried to isolate several immune cell types and sort of transplanted them intraperitoneally and followed by the susceptibility testing protocol. So from the point of view of traditional transplant immunology, it would be isogenic transplantation. And in fact, we found that dendritic cells from the susceptible offspring actually convey the phenotype, so that if you inject them into a normal mouse and then give them a small amount of allergen, they are positive for asthma-like features in this model. Now, other cells did not do that. Dendritic cell depleted fraction did not do that. CD4 cells isolated, or macrophages isolated, did not do that either in various quantities. So we seem to think that the effect is conveyed by dendritic cells. And here you're looking at BL eosinophilia in recipients in a testing with this low-dose protocol. And in fact, those that received dendritic cells from either asthmatic mother, here in the middle, and from DEP, diesel exhaust particle exposed mother family offspring, they all showed elevated BL eosinophilia, but those that received control, normal dendritic cells, did not have a change. So we looked for quite a while into what is it in these dendritic cells that is changed if you're born to an asthma or a particle-exposed mother. And for a long while, we couldn't find anything for as long as we were doing hypothesis-driven search. But when we looked into epigenetic profiles in these dendritic cells, we found, in fact, that there's a number of changes in DNA methylation that are seen both in allergy and particle-exposed offspring dendritic cells, and they are not seen in normal control. So we tried to link this epigenetic change to expression, and as I pointed out before, we couldn't really find expression changes at first. But then we hypothesized these changes are dormant, and they only appear when genes are activated by allergen exposure, for instance. And in fact, this seems to be the case. So there's very little transcriptional change in microarray assays in normal naive mice, but after allergen exposure, the change is quite substantial. And many of them overlap with methylation changes detected in the methylome array. So it would appear that the epigenetic change seen in these somatic dendritic cells comes at birth, and it is dormant until there's an allergen encounter, upon which it does its job in aberrantly regulating transcription. Well, this is a simplified way of looking at it, but many people ask, so what is the direction of change? What is better and what is worse? So in studies by others that I've shown before, and in our study, it appears that demethylation is generally better. And although if you look into detail, there's quite a lot of change in either direction, both in the hypermethylation and demethylation. And we don't really have a way to dissect these differences, but it would appear that demethylation might be better for you when it comes to dendritic cells. So we've designed an experiment to test this in vitro using a drug that induces genome-wide demethylation. We cultured dendritic cells from asthma at-risk offspring versus controlled dendritic cells, And they're shown here on the left side, and the yellow circles depict these aberrant methylation marks that we think are present there. And we co-cultured them with T-cell responders from a transgenic mouse where all T-cells respond to ovalbumin presentation by proliferating. And in fact, we hypothesized that, that the aberrant or the asthma at-risk dendritic cells would induce a stronger proliferation of T cells in response to ovalbumin, and this sort of mimics this feature of critical decision-making in the antigen presentation point where dendritic cells in diseased individuals for some reason decide to present the allergen, and in naive individual or in normal individuals this does not happen. And so we hypothesized that we can see this in vitro in a proliferation assay, and in fact we were correct. And it appears that dendritic cells from asthma at-risk neonates have a higher ability to present antigen to T helpers than control. But after treatment with a DNMT inhibitor drug, these features sort of goes away, becomes normalized, and now we don't see any longer this elevation of antigen presentation to CD4 T helpers. So getting back to the concept of transgenerational, we'll try to find out in mice whether asthma risk due to environmental particles is associated with transgenerational transmission of the phenotype. And for that, we aim to test for increased susceptibility in all the way to F3 offspring after a single maternal exposure to diesel exhaust particles. And then we'll test them in our low dose or intentionally suboptimal, as we call it, allergen protocol. And we'll look for allergic inflammation and elevated airway responsiveness. We will also isolate splenic dendritic cells and look for DNA methylome in all these generations to see if they persist or wane. And in the end, we'll try to drug intervention in vivo to try to abrogate this transmission and see if it changes anything. So we designed a model of transgenerational experience, transmission and for our experiment, and it's really sort of simple. It is the same cartoon as before. As you see, we have a particle-exposed mother and control, and we're looking at a susceptible at-risk neonate. It can be tested in our low-dose protocol. And we would let some of the females grow intact without any exposure and mate again to normal males and look into their offspring in a similar way. And then another portion would be also intact here, leading us to another group of mice to test. So now we have our F1, F2, and F3 generations for transgenerational testing. And in a subset of experiments, we'll try to inject a DNMT inhibitor drug in the F1 generation females to try and abrogate or reshuffle somehow the epigenome in the organism and see if it does any change. And in fact, it does seem to work, and both the airway inflammation and airway hyperresponsiveness transmit all the way to third generation. So in this chart on the left, you will see BAL as an epiallele in percent or counts, and the left bar is negative control. The middle bar is our main study group, exposure to diesel exhaust particle, and the positive control is a very strong ovalbumin-induced asthma control. So in F1 and in F2 and to a lesser extent in F3 generation, we see elevation in the diesel-exposed group, suggesting that their responsiveness to ovalbumin is similar to an asthmatic. As you see in the F3, I'm showing 4 liters separately because these liters were not as consistent as previous generations, suggesting that in some cases the signal may wane and in some cases not. And I would say that this is sort of what you would expect in an epigenetic versus genetic transmission. In a similar way, airway responsiveness to methacholine, shown here on the right, was also elevated in the diesel exhaust particle groups. This is F2 on the top and F3 on the bottom. And again, I'm showing liters separately, and you see they have some variability. Another agent that we try to use in the same protocol to try and trigger this transgenerational transmission is CAPS, concentrated urban air particles. And so far we have data till F2 generation, but in 2 separate cohorts, we have been able to see similar features in the phenotype, suggesting that in fact, just the pregnancy exposure to urban air particles can predispose to asthma risk in the progeny. And for the experimental treatment with DNMT inhibitor drug, showing here in both F2 and F3 generations, when the mother in F1 was treated with the DNMT inhibitor, there seems to be attenuation or disappearance of the asthma risk in this offspring, suggesting that our hypothesis about some reshuffling of the epigenome seems to interrupt this transmission. That's how we interpret it. But there's a couple of problems with this, and we'll focus in a minute on some limitations and some potential alternatives in future ways. But for now, from these studies by us and others, and in general in this paradigm to support the idea of transgenerational and epigenetic transmission, we could make the following summary. That in allergy and asthma, there are vast epigenetic changes. They are seen in people and they are seen in animal models. If you induce a change in DNA methylation, you can promote asthma in an experiment. We know that maternal exposures increase asthma risk, and this can transmit through several generations down. We know that epigenetically altered dendritic cells play a role in this, and we now sort of headed in the direction where we can show that epigenetically active drugs can abrogate the transmission. But one of the problems with this later approach is that epigenetically active drugs are A, quite toxic, and B, they're not detailed, so they're mechanistically not very helpful to us in trying to change the epigenome. Some of these drugs are approved for human use already, particularly in oncology, but it doesn't appear that they'll be sort of immediately applicable in asthma mechanistic studies and in asthma clinic. So what would be the way to try to direct these studies towards potential experimental and therapeutic agents? And one way to do this, we think, could be epigenetic engineering, sometimes also called epigenetic editing. So I'm shifting gears a little bit now to try to show what can we do about all this concept with epigenetics of asthma and its transgenerational features. Well, epigenetic engineering entails targeted ways to change an epigenetic set point of a gene or a group of genes, and to basically see a somewhat inheritable or stable effect. These agents could enable or disable transcription, and my personal interest is with enabling it because I think it's more interesting to me. There are other ways to disable a gene in the genome, but there isn't a way to reactivate an epigenetically silenced gene. Of course, such approaches would be best if they are targeted, either to a specific gene or to a small group of genes. You could potentially expect a heritable, at least mitotically heritable effect from a single entry of your agent, as opposed to overexpressing or administering a recombinant protein. And you could potentially avoid the need for transduction or other viral vectors, which is one of the important obstacles towards therapeutics. So we've designed this concept and tested it in a gene that doesn't really have immediate relevance for asthma, and we've picked it based on information about its sensitivity to methylation in the promoter. But the proof of concept studies are as follows. We have a fusion molecule that has a DNA binding domain and an epigenetically active enzyme, and we express it in a cell in a way that this domain is designed to target this particular gene sequence. So the hypothesis would be that if we have this demethylase, depicted here by this red cucumber, the promoter would become demethylated in the vicinity of this enzyme, and it would now be open for transcriptional stimulation by, for instance, interferon and STAT pathway. But in a control scenario where we use either a catalytically inactive mutant enzyme or none at all, the promoter would remain methylated, there cannot be a stimulation, and there won't be transcription. So we use, for DNA binding domain, zinc finger protein arrays. And these are proteins that can bind to DNA bases sequence specifically. There are other motifs that people now use, including TAL effectors, and some people are trying to use CRISPRs for epigenetic purpose. And these are fused in a protein with an enzyme, or prototypical enzyme, so to say, putative. One is called TDG, thymine DNA glycosylase. Another one we're trying is called TET, 1011 translocation protein. And these are the enzymes that apparently work together to demethylate DNA. And we express them in a cell where they are produced as proteins and bind as promoter. And in theory, we're trying to produce them as recombinant proteins and treat them in culture to cells, but we're still working on that. And long story short, and I don't expect you to look into every detail on this slide, but in general, in this study, we found that this protein shown here, as opposed to a mutant catalytically inactive protein, the control would be in the middle, or a zinc finger alone would be in the left. It does lead to a drop in methylation in the promoter of this target gene where we wanted to do this in a number of sites. And this is associated with increased transcriptional response, for instance, to LPS and interferon, shown in the graphs on the right. And this is also associated with increased production of the protein by this enzyme that we're demethylating. So one of the key questions in this area is how specific can this be? And in fact, there are some limitations and some tools to overcome these limitations when it comes to design of these DNA binding domains. And I don't need to go into the details of this perhaps, but we've tried a microarray assay in the cell types to see if we've affected other genes than the one we're targeting. And we've come up with a list of 40-something genes that changed expression as well. But when we did pathway analysis, we found that most of these genes are connected to our target gene as the one that we're trying to elevate. And in fact, it is elevated. And so we think these changes in these genes could be secondary to our target molecule. And there's only 4 molecules that were changed in transcription that were not linked to this target gene NOS2. And so we think it's not a very bad specificity if you're trying to use this, for instance, in an experiment. So, I think that epigenetic editing is a promising, cutting-edge technology. And it can have a variety of potential applications that could be seen both in asthma and in pulmonary disease and outside, broadly speaking. You could enable a gene of interest in a particular cell type or a site in a tissue or an organ, so you can confer unusual properties to the cells, for instance, for immune enhancement or hormone production, because all the genes are already there. All you need is to activate them. It can be potentially used in transdifferentiation studies, turning one cell to another. It could be used as supportive tools in stem cell regeneration medicine. And importantly for us, it can also be a helpful causality tool in environmental and in exposure studies in epigenetics and asthma. And so I think we might come in the future to something that we might call an epigenetically engineered cell. And this cell would be treated through these type of agents and it'd be native to the organism. It would be genetically intact, but it would have a personalized, controlled, and heritable transcriptome, a sort of a custom cell. And I think this is an exciting concept. So this is basically all what I have for today, and I'd like to acknowledge people who work for me, Dr. Gregory, people who worked for me in the past, Dr. Mikhailova and Yiming Zhang. And I'd like to thank my colleagues, Les Kobzik, Caroline Owen, Ivan Rosas, and Bart Shelley. And our studies are supported by NIHS. Thank you very much. That is all. Any questions, please?
Terrific. Thank you very much. Here's a question.
So you showed some pretty convincing evidence of the role of epigenetically perturbed dendritic cells in this response to the allergen. But how certain are we that the dendritic cells are holding onto the memory? Like when you were showing that by inhibiting the DNase, the transferase with that inhibitor, that the transgenerational effect was cut off. Do you know that inhibitor was targeting just specifically the dendritic cells, or is there potentially some global effect where the allergen responses through the dendritic cells, but the idea that the dendritic cells should be easily responding to that stimulus is sort of held elsewhere, and perhaps is that related to your epigenetic engineering, where you would be specifically trying to sort of target the dendritic cells in terms of fiddling with their demethylation?
Can you try and repeat the question?
Right. Well, I understand that the question is regarding the non-targeted way that epigenetically active drugs act. And it has something to do with us showing epigenetic change in dendritic cells, and we're trying to show this in the transgenerational study. And we're acting on these mice with an epigenetically active drug, but we don't know if the change is primary to the dendritic cell, or does it also occur somewhere in the organism and affects the cell in a secondary way. And I think you are correct. We cannot dissect one from another, because these drugs have organism-wide effect, and they also have epigenome-wide effect. So there isn't a way to pinpoint this in vivo and say, well, we have affected this particular cell type and this particular set of genes. We're trying to do this in experiment when we do this in vitro co-culture studies, where we're only affecting the dendritic cell because it's the only cell in the dish. And it does seem to change somewhat an outcome that is characteristic in asthma. But again, there's no guarantee that this is what happens in vivo, and I'm sure you're right, and there's probably a lot more things happen to the immune system when it is exposed to this drug. Now, as to the future of whether we can use targeted epigenetic tools for this, the future will show. Hopefully, yes, but I can't say how.
Can I ask, in your model of the transgenerational transmission, are F1 generation, F2, F3, are they all female mice? And I wondered if you had looked at male mice, whether they would inherit some of these epigenetic changes that could then be passed transgenerationally.
Well, the outcome of allergy susceptibility, when we test this, this is not gender stratified. So we include both female and males. But the transmission, we know it is occurring in a female line. And we've looked into some control experiments, and it doesn't seem that paternal line would transmit that. So eventually we're looking into the material as well from the female mice in these cohorts because we're focused in this area.
Just help me then. So does that mean that the implication of the epigenetic change is that the ovary is more susceptible than the testes for transmitting these, or not so?
Well, we don't know. I mean, there are some studies that are not asthma related that show paternal transmission, and in fact paternal transgenerational transmission as well. So male germ cells are probably as susceptible as ovary, or more studies are needed. But because asthma has a maternal history where we're looking into this particular gender, although it's fair to say that in some other cases it doesn't have to be gender specific.
So I had one more thing that I'm trying to understand the implications of what you've taught us for my understanding of evolution. And this possibility that epigenetic changes might influence in some way the course of evolutionary change. And I wondered if, I'm trying to imagine the volcanic eruption and a species exposed to the particulates from air pollutants, not urban air pollutants, but environmental air. Is there any suggestion that genetic mutations evolved in sudden stepwise fashion after some, sorry, it's a way out question, I'm sorry, but I'm trying to incorporate how this might impact other species' evolution over time.
Well, it's an interesting concept, and you sound like Lamarck. So for epigenetic phenomena, it is unclear whether it's played a role in evolution or not, and this is what actually Lamarck implied, but there are some experiments, and it's a long story of neo-Lamarckism. But it's kind of hard to say because some of the experiments that were heading in this direction didn't really get validated by others. And it is quite rare, and I don't think it's well described, at least in mammals, that an epigenetic change once introduced would be permanently inherited. And so that it would become a source for a new species or variegation. But you're right that I think in the history of evolution, larger catastrophes act as a pressure factor in the natural selection. And I don't know if they would act as a factor in epigenetic mutation, but in theory, this is possible. Whether this would lead to a change in species, there's no way of saying.
All right, one last quick question, sorry. Would you give folate to your pregnant wife? Is this a non-specific methylator? And there are clear neurologic benefits, I gather, to folate in pregnancy, but is there some concern about a non-specific methylation of the genome?
There is, and there's some concerns about folate supplementation goes beyond the epigenetics, and it is a debatable area now. And it would appear that in high-risk environments where there is a risk of folate deficiency that is well-founded, this can be justified, but otherwise perhaps not. And there isn't a consensus as far as I know, but you're right in suspecting that this can have deleterious outcomes.
Wow, fascinating material. Thank you so much. Thank you all for participating. See you next time.
Thank you very much.